Hello, everybody. In this lecture, we're going to talk about the extracellular structures of eukaryotes. So let's get right into it. So there are a bunch of objectives here. By the end of this module, you should be able to discuss the basic structural and functional features of the plasma membrane, um, give examples of eukaryotic cell walls and how they differ in the kingdoms, describe the eukaryotic glycocalyx and list some of its roles. Discuss the structure and function of eukaryotic flagella and compare them to prokaryotic flagella, and describe the structure and function of cilia. All right. So all cells have a plasma membrane. So we talked about that with prokaryotes. It is the same with eukaryotes. So all eukaryotic kingdoms, we're going to have a plasma membrane. This is pretty much a defining feature of a cell. Now, one of the major differences between the eukaryotic membrane and the prokaryotic membrane is the presence of sterols. In animal cells in particular, you have the presence of cholesterol. And cholesterol is something that helps maintain optimized membrane fluidity. So it helps under cold conditions to keep from becoming too rigid, and it helps under warm conditions to keep from becoming too fluid. So this is an extra tool in the eukaryotic tool bag, I guess, to help maintain membrane fluidity. Now, just like a prokaryotic membrane, it's a selective barrier. It is going to have a fluid mosaic model, just like a prokaryotic membrane, and it's going to have that phospholipid bilayer, just like the prokaryotic membrane. Now, many, but not all, eukaryotes contain cell walls. So animal cells do not have a cell wall. However, in your fungal cells, you're going to have a cell wall that's made primarily of chitin, which we see here on the right-hand side. They do not have peptidoglycan. The cell wall is going to function similarly for all organisms, whether they're prokaryotic or eukaryotic, in that they help maintain cell shape and they protect against mechanical and osmotic stress. And so here we have the fungal cell wall. We're just going to kind of go quickly through some of the different types of cell walls because there is a bit of diversity here. The fungal cell wall is primarily made up of chitin. We also have this beta-glucan in here. When we go to our plant cell wall, this is the most abundant biopolymer on the planet. That is cellulose. They also have some hemicellulose in there. And then we can go to algal cells where we have some varying polymers. But for example, here in these green algae cells, we see that they are also made up of cellulose and hemicellulose. But then you get some real like wild things. And diatoms have silica membranes. And so they actually have inorganic silica around the outside of their cell, which makes them very pretty. So there are some other external structures that eukaryotes have. They play roles in protecting the cell, cell adhesion, and motility. So if we start out with the glycocalyx, this is similar to the prokaryotic glycocalyx. This is an outer layer outside of the cell wall, outside of the plasma membrane, and it contains primarily carbohydrates, some glycoproteins, and glycolipids. Keep in mind that glycoproteins and glycolipids are proteins and lipids that are modified with sugars. And so, that's where the name glycocalyx comes from. And it's going to have a function similar to that of prokaryotes. It's going to help with adhesion. It's going to help with cell-to-cell contact, stuff along those lines. Now, flagella in eukaryotes are interesting because they're quite different from flagella in prokaryotes. Now, functionally, they may have the same overall goal, but they're not exactly working the same way. So the big difference is your prokaryotic flagella, as you know, is made of flagellin protein, while in eukaryotes is made of tubulin. which make up microtubules. Those microtubules in the flagella of the eukaryote is a 9+2 arrangement. We'll see that in a membrane in a minute. But another thing that is interesting about them is that they are completely membrane enclosed, whereas in your in your prokaryotes, the flagella just extends out through the membrane and does not actually get enclosed by the membrane. The flagella of eukaryotes are going to be connected to microtubules, and they have a wave-like motion rather than the rotary propeller motion of your prokaryotes. And so when we look at this, we see here these microtubules, they're going to be connected to a centriole, and they are going to have this 9+2 arrangement. So you get 9 around the outside, and then you get 2 on the center. And so that basal body that they're anchored to on the inside is your centriole. Now they have this wave-like back and forth motion that allow them to swim. And so this is the opposite of, or not the opposite, different than prokaryotes, but actually rotate like a propeller. All right. And then another type of structure that's used for motility in your eukaryotes are cilia. And these are structures that are similar to flagella, but they're usually much smaller and more numerous. So there are a lot of cilia on individual cells. Now, another big difference is in how they move. Now, you've got this wave-like motion that you get with your single flagella, but with the cilia, what you're going to do is you get kind of like an oar-like motion. And so this has this power stroke, recovery stroke back and forth. And so it's this, think about if you've ever been in a boat or a canoe and you've been going through and rowing. This is the same sort of idea that's happening, or the same sort of motion that's happening with these cilia. All right. And so that is a brief overview of some of these extracellular structures. You should now be able to discuss the differences and similarities between the membranes and the cell walls, as well as discussing the functional and structural relationships of the glycocalyx, the flagella, and the cilia. And if at all possible, try to make connections to how these relate to prokaryotes, because there are some differences in there. With that, thank you very much, and see you soon.